Hello to everyone joining us to watch or listen to this conversation. I am Dr. Edith Brignoni, and my mission is to share critical evidence-based information that helps improve brain health for as many people as possible. Today, I have the honor of being joined by Phil Newsome, a neuroscientist in training whose work sheds light on one of the most fascinating and often overlooked aspects of human development, the fatherhood brain. Welcome, Phil. How are
Thank you for
you?
having me. I'm
Of
good.
course.
I'm good. I just told you I was running from class and I made it here, so I'm happy to be here.
I'm happy to find a break or distraction or positive distraction from your coursework to share with me and our community the amazing knowledge that you have on the plate, on the table for us today. So to begin, I'd love to hear a bit about you as a person and as a scientist. So to start with a question, I would love to hear what life mission are you on right now and why?
Yeah. So what my life mission? That's a big one to start off. I mean, I think essentially my interest in psychology stemmed from really high school. I took a AP psychology course that I really enjoyed. Um, and when I went to undergrad, I wanted to kind of continue that search for that. And so I studied psychology and biology. And I think beyond undergrad, it really, like, I think I just had a shift of, like, I knew I wanted to go into research. I was really fascinated by the things that we didn't know more so than the stuff that we did. And I think, applying skills from multiple disciplines to uh, reveal things about the human experience is just something that, I think is incredibly important and what I want to do really for the rest of my professional life.
That's exciting, and it's a wonderful mission both for you individually, because I believe it will always keep you in a pathway of constant learning and therefore growth. And at the same
Mhm.
time, it's even more wonderful that your personal mission, your enterprise will lead you to help many, many other humans as well. So in 2025, aka this year, you published a peer-reviewed paper in the journal Human Brain Mapping titled My Baby versus the World, Father's Neural Processing of Own Infant, Unfamiliar Infant, and Romantic Partner Stimuli. It's a study that, at least to me, really captures something profound about how fatherhood reshapes the human brain, which is not something that we talk often. So I'd love you to tell us what main question were you and your co-authors trying to answer with this study that ultimately got published? And where did that question come from? For example, was it inspired by prior data in your lab, an observation from someone, or perhaps a deeper curiosity about caregiving and emotion? And before diving further with this question, also we will explore your academic journey in relation to it, but let's start with the question and how it got inspired.
Yeah, yeah. So, um, for about 2 decades, or a little over 2 decades, neuroscientists have been interested in how parents respond to their infant, whether it be in the brain or otherwise physiologically. And most of this work has been done in mothers. Um, so there's been, I think, probably less than 10, maybe a little bit more, but probably less, of people that have studied fathers' responses to their infant in the scanners. So we were really interested in kind of extending that literature. One of the biggest limitations to this literature is that we're oftentimes contrasting own infant responses with something like unfamiliar infant responses. So we'll look at how the brain responds to their own baby versus some other baby. Um, and we were interested, was like, that's cool, but it could just be the fact when they have unique responses to their own infant, it could just be due to the fact that they're looking at like a personally familiar stimulus that they love and care about. So in our task, we added a romantic partner stimulus, which was going to help us kind of really pin down if there was a unique response to the baby, above and beyond familiarity and infant features.
Thank you for that response. To me, that really contextualizes predominantly the scientific aspect of the question and therefore the study. But I want our audience to also learn how science, we like it or not, or not we like it or not, but is it explicit or not, as much as, of course, it builds from other people's work. It does have a deep root on actual needs and experiences and pains of the human experience. So I'm not sure if you're directly connected with the scientists that originated this question and why. So if you can also share with us a little bit of that non-scientific aspect that promoted science to actually test the question and all the other aspects we're gonna discuss over that.
Yeah, it's a great question. So I think in general, I was mentioning that a lot of the work has been really focused on mothers in terms of the parenting brain literature. And I think that comes with like this kind of social concept of kind of mothers being the parent and then the fathers being somewhat of an optional like parent figure, which is I think in recent years, we're seeing increasing rates of fatherhood involvement. So I think the work that we're doing in our lab, which really focuses on the father, the biological transition to fatherhood, is trying to sort of line up with the fact that fathers are also going through significant changes and they deserve to have research that gets at their experience and hopefully leads to better support and policies that can kind of help them make that adjustment.
That is great because you guys are highlighting a critical aspects of caregiving experience, both for the child, but also the caregivers, let's say mother and father. And as you highlighted, the niche in terms of scientific field studying what happens in the human brain as a function of raising kids, it's still relatively new. And if anything, in mothers, and even newer, the other part, which is the father, and how does that relate both for understanding the father's brain and supporting them in their own brain health and parenting journey, but as you said, to make sure that the mother is not left off as the sole responsible of that experience. And we make sure that both parties involved in that experience, if that is a nomenclature, if this applies to same-sex couples, to heterosexuals, but it's the role themselves of fatherhood and motherhood in being in complement. Now, diving a bit deeper in also understanding who you are, what is the individual component behind this study, you primarily as the first author, that for context, to our audience that may not be too familiarized with science and how studies publication work, but we can assume that as much as this is a team effort, in this case, Phil predominantly led the A to Z, if you will say, in collaboration, of course, with all the co-authors in this paper. And I would love that you share with us that academic journey of yours, if you will say what from high school brought you into whichever undergraduate program you joined. You touched a little bit on that, but if you can say again, why back in that very younger mindset of yours, transitioning from high school to the university world, and how the long-term projections that you have from there, if any, mirror what you have right now versus not, and also how research was presented to you in that journey, that for some is obvious and you had the great opportunity to know about it as a career from the very beginning, even from school time, and others is just as you stumble around or meet people or find opportunities. So tell us about your journey.
Yeah, absolutely. So kind of, as I mentioned before, I had taken a psychology class in high school and ended up really liking it. And I feel like high school was kind of a perfect time to have a psychology course because the high school, I was like a senior, the high school to college transition in itself is such a salient experience. And we had talked a lot about in that class about how these kind of major transitions that happen over the lifespan kind of come with these not only psychosocial changes, but also biological changes as well. So that was something that was kind of implanted in me from the beginning of my psychology journey. And then when I went on to undergrad at St. Mary's College, I knew from the start that I was going to do psychology and biology like that. I didn't waste time starting my studies. And just, I think throughout that process, I was fortunate to have teachers that really cared about teaching. And I learned a lot of stuff about developmental psychology and psychophysiology and biological psychology that really ended up shaping kind of like where my research program ended up going. Importantly, my undergraduate institution was not an R1. It was a smaller liberal arts college. So mostly the experience I was getting there was kind of in the classroom. And I just happened to have this one professor, Dr. Kathleen Talent, who's no longer at St. Mary's, but is in private practice. I was able to work at her private practice over the summers to get some extra experience and exposure to kind of hands-on psychology clinic work. And I think that brought the classroom kind of to life for me in seeing the kind of the importance of the things that we were studying. And then from St. Mary's College, once I graduated from there, that's when I really marked the start of my research career, like right when I ended undergraduate. So probably a little later than some people. But just based on the clinical experience I got in undergrad, I thus became interested in what didn't we know? Like, I knew some of the stats about, CBT for anxiety is only working at 60-65% at the time. And it just seemed kind of egregious because we wouldn't see that stat in many of the physical health aspects. So it was just kind of disheartening that we had made progress, but it didn't seem like we were good enough yet. So I got to Maryland, which was where I did my master's, and I started cold emailing PIs because I was trying to look for some research experiences. And I think I cold emailed like 30 PIs, principal investigators for the viewers, the directors of the lab. And one person responded, and it was Dr. Alex Shackman. And I ended up getting into his lab as a volunteer for a semester, and really got some amazing experience. And we were studying the nature and neurobiology of fear and anxiety, which is really where I started my research career, is in that space. And then from there, I moved on to a 2-year post-baccalaureate fellowship at the NIH with Dr. Daniel Pine, doing kind of similar work, but I was really focusing on kids. So at that point, I really started to integrate my interest in development, developmental processes, and affective science and biology. It kind of all came together when I got to NIH, and ultimately led me to kind of where I am now in studying kind of another very salient transition period across the lifespan, which is parenthood.
Thank you for sharing that for the audience, because that's precisely what I want them to see, to learn that no pathway to scientific endeavors are the same, mainly at those initial stages, transitioning from high school to undergrad, and then depending upon where you're in the world, as I've spoken with other invited guests that have spoken in this channel, you may be in the US, you may be in Europe, you may be in a country in another region of the world, and the education opportunities vary. And also the opportunities to do or not do research, and whether it's volunteer versus paid. And that's where I wanted you to share with us how maybe you line up 10 different developmental cognitive neuroscientists, and they all have their own way to have gotten to being co-authors, for example, in a paper like yours. And interesting that you did the work in Dr. Pine. I'm not sure if you got to work with adolescents or irritability. I believe that that's one of the topics they do touch in there. But that was one of my questions, like how ultimately you end up being interested in the neuroscience of fatherhood. And I will let you to expand on it if you want to, but if I can rephrase for the audience what I've learned from your journey is that a lot of what you're doing right now, topic wise, methodology wise, and training, or how you got informed to decide which pathway in terms of training to take, was very much in foundation motivated by your own intricacies, your interests, and choosing an undergraduate pathway in psychology and biology. But after that, really letting yourself being exposed to opportunities, but also being very proactive. I love how you said I called email about 30 PIs,
The
principal
whole
investigators.
department got an email from me.
Yes, and that is the move. That's what I tell mainly to young scientists, to young students. No matter if you will be scientists or not, I believe this is a learning, not only for younger, for at any stage in our lives, even more if we're pivoting from fields, from topics, the value of, you know what, if you really know that you want at least the opportunity, call email. What is the worst thing that can happen? That someone answers. Great. So maybe if one from 30 happened, look, you had an amazing opportunity. Then you did your not only your research, but that led you also to the NIH and so on. These were building blocks for providing you later the opportunity where you are right now as a third year PhD student, really deep diving and fine tuning your critical thinking skills and all the knowledge and skills that come with a PhD training that, as I tell people, to me personally goes beyond even the content that you will be learning, but because science is constantly evolving and changing beyond the content, which probably in 5, 10 or more years, it will be coming away obsolete and you have to keep updating yourself content-wise. It's about all the skills that you developed and how you understand how to test questions from the world. And in the context of fatherhood, please confirm now that I pause, but I believe that it has been a trajectory, as you said, that you may not have initially planned to be in the field of developmental cognitive neuroscience, even less parental, like parenthood in general, and even less the niche of fatherhood and brain, which is just starting in a way. And that's great because you and your co-authors are pioneer of a wonderful field. How do you feel that that transition happened, and was a balance between opportunity versus that you genuinely need to like the topic, regardless if it's your experience or not?
Yeah, great question. So I think, yeah, the main theme really is I got really interested in these like really large transition moments across the lifespan because they seem to be not only a time for great joy and happiness, but also a time for heightened risk for psychopathology. And so that's kind of where I just placed my focus broadly when I was like applying for graduate schools. And I think when I came across Darby's lab and we got to talking a lot about it, there were so many things that like I was thinking about in Danny's lab in terms of what's part of the etiology or the pathway to or development of anxiety, like what's the parents' role in that kind of development. Whereas Darby was really focusing on, well, let's look at parents first and their health and how they're making this transition to parenthood. And maybe then that will tell us something about not only their long-term health beyond that, but then their child's health as well. So it kind of actually was a really nice kind of two-sided thing where like I had this experience working with children who are highly influenced by what their parents are doing. And now I have this experience to really do a deep dive into parenthood and fatherhood specifically, which is clearly influenced based on some of the work that we've done by what their children are up to and how they're interacting with their children. So I feel like the transition to graduate school and studying fatherhood, it just felt natural because I felt like I was going to get like this other context for this more broader kind of goal that I had was just studying these major life transitions, the effect of neuroaffective experiences across major life transitions. Yeah.
That's great, because it's a mix of, I believe, you as a scientist, as an individual, finding in the context of opportunity and in relation with other more senior scientists, where is your niche, both interest-wise as well as training that may or may not be applied in the context you are right now, which is the academia, and how ultimately this research, as you said, it's one of the many experience. As you said, you were very interested in how across life these windows or experiences can change, and that I resonate a lot with that. I ended up for my PhD in a lab that primarily was focused on learning abilities, which is a big umbrella in itself. And I was coming with a strong interest on what are the roles, the impacts directly in brain function and our structure of early experiences. And that, again, is another big umbrella. It sort of resonates with what you were saying. And among the many different experiences that we can understand, one of the many in the context of learning that I decided to tap into is in the context of early dual language use, and how that in itself connected both with what the lab was already doing, even if it didn't involve bilingualism or dual language users for learning abilities. But I brought the interest of early experiences, and I want our audience to understand that in science, a lot of what gets produced and tested and the team efforts is I will say, it's a dance. It's a dance of people's interests, funding opportunities, and key questions that are out there, and how we do these synergies, both when we have senior scientists and the incoming students like you, or fellows like postdocs. So doing segue to deep diving into your recently published paper, I would love that you share with our audience the study design, that you describe for us how the study was designed, for example, what stimuli fathers viewed, and what comparisons you made to address, to test your question.
Yeah, so just very briefly, the study pulls from a larger longitudinal sample of fathers that we've been collecting for over 8 years. We started in the prenatal period, and now those kids are 7. So the data that we used for this particular project was specifying that early postpartum period. So they got scanned about 8 months after the birth of their first child. And what we were, as I mentioned before, what we're really interested in is the unique neural responses to their own infant. And could we predict anything about their bonding experience, or how their affective experience, how stressed they were during this time based on these responses? And what we found, at least what the main results are, is that we found that there was these heightened activation to the own infant versus an unfamiliar infant, and predominantly in regions that deal with mentalizing, which essentially are regions that support the ability to interpret and understand others' thoughts and feelings, put yourself in their shoes. Perhaps it's kind of like a precursor to empathy, if you will. We also saw heightened activation in regions that deal with reward and emotion regulation. When we compared the own infant responses to their romantic partner, we found that there's an activation in this large cluster in the precuneus, which is a posterior midline region of the cortex, also kind of dealing with mentalizing, higher order cognition, social cognition processes. So those were the initial findings, and I don't know if we want to pause there and talk about that, or I can go into some of the other stuff too.
Yeah, we can break down in a back and forth manner with key questions I have to really dismantle and walk our audience step by step in different areas of the key findings and the interpretations of them. For example, you mentioned the brain findings. What specifically in terms of patterns of brain activity really stood out for you guys when fathers saw their own infant, for example, as compared to unfamiliar infants or partners. And also another question I had is which networks were most involved? You mentioned empathy at some point, but in the context of, for example, reward, salience, social cognition, were there networks that were more involved or not? And in which comparison? I guess at the statistical comparison level, which is what I tell people ultimately matters beyond looking side by side.
Right. So yeah, I mean, the things that really stood out were, one, a lot of the regions that we saw that lit up, quote unquote, when we looked at when fathers looked at their own infant versus an unfamiliar infant, were very similar to what we actually find when mothers look at their own infant and unfamiliar infant. And it's predominantly these regions in the cortex that are dealing with social cognition. We also have things thrown in there like the OFC, or orbitofrontal cortex, which is nosebleed supports reward. We also have some emotion regulation regions thrown in there as well. But largely, we see this kind of social cognition network that's really at work when discerning between your own infant and an unfamiliar infant. And then, kind of like I said, the novel contrast when we're comparing the own infant responses to the romantic partner responses, it was that large posterior midline region that, again, is really supporting social cognitive processes.
Wonderful. And I had also a question that it's probably tapping more into interpretations you guys may or may have not done. So we'll learn now. But it's in this contrast of own infant versus unfamiliar infant. I wanted to know, how did it reveal, if at all, something about bonding and or caregiver motivation in this father sample?
It's a great question. So actually, the second kind of round of analyses we did was we wanted to compare brain responses for our different contrast with some of these kind of out of brain measures of parenting. So we looked at the father's bond with their infant, number of bonding problems, parenting stress, total time spent as primary caregiver, things that we know are kind of like indicative of the parenting experience as a whole. And we found that that posterior midline region of the cortex, the precuneus, and actually the posterior cingulate seem to be significantly associated with most of these things. So in the positive direction, so just to clarify, increased activation in this posterior midline region, we're looking at your own infant versus an unfamiliar infant, was positively associated with father-infant bond. So if the father was more bonded with their infant, or at least reported that they were more bonded with their infant, they had heightened activation to their own infant versus an unfamiliar infant. And we saw the opposite direction when we looked at something like parenting stress. So we saw heightened activation to this posterior midline region when looking at own infant versus unfamiliar infant. And this was associated with lower parenting stress.
Wonderful. That's where I wanted us to deep dive. And it's more of a follow-up methodological nerdy question, but I think it's critical.
My favorite.
Yeah, it's I don't know if you find it nerdy enough, but at least when you were mentioning the different ways in which you assess anything related to bonding or caregiving, I got very I don't want to say shocked, but curious when you mentioned, I think it was bonding times struggles or problems. You said something along that line. How did, just out of curiosity, how did you measure that? I assume it's self-report would be my guess, but even in the self-report, how is your instrument, or what did you characterize to then people then self-report such metrics?
It's a great question. So yeah, we actually had two separate indices for parent bonding. The first one was like father-infant bonding using, it's called the PPNAS. And that is basically kind of like the positive spin on like how much did they feel bonded to their infant. And that's all self-report. Well, actually, I should clarify, all the out-of-brain measures are self-report. And they're all collected around 3 months postpartum. On the flip side, we had this measurement called the postpartum bonding problems, essentially. It's what it was indexing. And it's a similar measure. They're clearly like inversely correlated, these two measures, but still slightly different. Like the questions aren't just like inverted. There's a couple different questions, but it's really indexing how much struggle or how much problems there was, kind of like in terms of like feeling close to your infant.
Thank you. I love it because we will dive even deeper in the context of psychology, because I think that even more so in a world that we have oversaturation of information and people are struggling to tease apart what to believe or not, and even within science, what is reliable studies that are published versus not. And part of our education here as scientists is to also share with our audience the different ways in which we test questions, with also the limitations that we have at the experimental setup. And in the context of self-report metrics, I know that you're totally familiarized. Some people may question them, but that's why also there are lines of studies, like scientific fields in themselves, that validates these self-reports metrics and instruments. So later other scientists, other labs can use them. And there's nothing wrong in self-report, and they are also validated. They are reliable metrics for us to use together with other kind of data, including brain imaging data, to test different questions. So if you can speak to our audience a little bit on that topic, and even more so the specific instruments you guys used, were they internally developed, or were developed and already published by others and you just adopt and use them?
Yeah, great question. We're getting to the nitty gritty here. So the self-report measures we used were all previously validated for psychometric reliability and validity and all those things. They also showed really strong internal consistency in our sample. So they had been used before, they had been validated, they have been deemed reliable sources of information for these kind of things. However, your point well taken that I mean, the limitation with self-report is people can say anything, you know, so it might not be exactly indexing what we're getting at all the time. So the best we can do is, another word I keep mentioning, but the best we can do is really kind of look at the psychometrics and just see how good those come out and kind of base our interpretation on that. I mean, the other thing to say is I think there's also something actually really inherently important in just getting a subjective report of these things, even if it's somewhat separate than the actual thing. And there's been a lot of work on this about like how like the subjective report, let's say, of like anxiety or depression or anything like that, is actually incredibly predictive of subsequent like kind of like serious psychopathological events, maybe even more so than some of the more what we would quote unquote call objective measures, whether physiology or whatever. Yeah. So it's definitely providing us something interesting, even if you know, people might, you know, slip up and answer something wrong or don't understand the question, which happens obviously sometimes.
Yeah, thank you for being very upfront on sharing with our audience how us from labs, from science, and again, we have questions and we designed as best as we can with the resources we have, balancing, and even more so in human research, balancing the controls and curating that later indeed we can make claims of the associations of these variables, and at the same time keeping it as naturalistic as possible, as psychological as possible, so we can later extrapolate and generalize to the outer world. And that leads me to then also ask you, so you share with our audience in the context of methods, and why do I go to the nitty-gritty of methods? For our audience, they need to also understand that you and I, it's obvious, but again, when I tell people, before I even read your discussion, show me your methods and your results. Like, that's what I want to see, because that's where everything, the rest is a story. Which is good, and you put things in context, both in the intro and in the discussion, but it's very critical that for us to really understand given results, we need to look at the methods and which were your confounding variables, whether you control for anything, and if so, for what and why. Like, people even if they're not scientists, they really understand why these factors and why you control for them in the context of looking at both brain circuitries at the functional level with bonding or caregiving metrics in a fatherhood sample.
Yeah, it's a great question. Oh, I love this question. So, because it actually gives me a chance to explain that our sample.
I love questions, and I'm a nerd. So
no,
here we
I
go.
love it. So, our sample was actually collected during an interesting period of history, the pandemic. So, one thing that we were really concerned about going in when we're looking at these data is time between scans. So I know, and I reported this in the paper, I tried to be very transparent about this. I put like a little figure that showed there were some subjects that actually there's time between, or the time, sorry, not time between scans, time between birth and that scan, and that postpartum scan. And you saw that some fathers, the time between that their first child's birth and that second scan was a lot longer than most of our other fathers, simply because there was a pandemic and we couldn't get them to see us and into the scanner. So that was something that we not only controlled for in our analysis, which we found did not do anything to the results, which was, thanked our lucky stars. We also did a follow-up and kind of just ran a couple like simple like statistics just to determine if there was differences between that smaller group of dads that kind of like had this like longer space between the birth and that first scan, and the other fathers in terms of like demographics and some of their self-report measures. We just kind of compared them with like a t-test, and just to see if there was any significant differences up front. And we found that overall there was really no differences in terms of how they reported on a lot of the measures, and ultimately when controlling for it in the final model, it didn't do anything to the results. So, but it was a concern to your point.
That's great, and I'm glad that you both show to our audience here like the behind the scenes of what you deal with in terms of when you were ongoing and retrospective how you dealt with it. That including in the paper, you were very upfront, as we all scientists should be, on saying what are the limitations. We were able to address it in this way. And this is the extent of how far we can interpret versus know with what we got. And there's always time for newer studies and more, but at least it's a great foundation. And more at the population level, we know that key factors such as socioeconomic status and the different factors that compose that composite in itself. So either the whole cluster of socioeconomic status or some factors within it. Did you guys care at all about it? And or were there also data collection and therefore controlled in some covariates in a way about, maybe this is for another study, but fatherhood in the context of non-biological fatherhood? So anything that you can share with us about
so many great questions, and we could talk a lot about that. But the first thing, socioeconomic status was an important point you made. I think that was one of our limitations. Is, well, I think our sample is quite. We have a really we have a sample that's really representative of our area in Southern California, racially and ethnically, which I think makes our sample really strong. However, they skew on the higher end of the socioeconomic spectrum. So unfortunately, I don't think we have the data to really kind of determine if there's like kind of this SES effect kind of on our results. But absolutely, I mean, there should be future work that looks at this, and I know of some groups that are going to be getting into that and seeing like how like people that are like more disadvantaged financially, how that affects their development across the transition to parenthood. I mean, those studies are coming from some other groups, but this study wasn't able to really get at that. I'm forgetting that second point that you made, but it was important.
No, I was also talking in the context of non-biological fatherhood,
Yes.
and I think that will be for another study, because inherently, I guess in here, guys, I'll let you speak about it.
No, I wanted to make sure I didn't forget it because it was such a good point.
Yes. Yes.
Again, we don't have the sample. We didn't have enough people reporting of non-traditional, non-heterosexual relationships. However, go back to a study in 2014 from Ruth Feldman's group. I think it was led by Abraham and colleagues. It was in PNAS, or Proceedings of National Academy of Sciences, where they actually did do that study, where they looked at brain responses to mothers, brain responses from mothers about their own infant, to fathers that are in heterosexual relationships that were not the primary caregiver. They looked at their brain responses, and they also had a sample of fathers that are in homosexual relationships and were the primary caregiver. And what they found is that the fathers that were primary caregivers in the homosexual relationships, their brain responses were much more similar to the mothers to their own infant compared to the fathers that were secondary caregivers, providing some evidence, some pretty compelling evidence that there is a experience-driven biology to parenthood that extends beyond kind of the hormonal fluctuations that comes with pregnancy.
Very, very interested. And I know that some colleagues of yours, more from the maternal brain side of things, and you all complementing each other, are also, as you all move forward with foundational studies, will move forward with populations of the same or similar experience, but different backgrounds. And in here, when I make these questions, I don't want you, and even less our audience, to interpret them as challenging the study in itself, but the opposite, to demonstrate for our audience, I know that you're clear on this, that again, science is building blocks little by little. We do this step, then the next one, and the next one, and they inform each other. It's a long journey, and we just start putting research out there that was critical as new questions will arise, both that you already had them in mind or just from the results you have. And wrapping up with the key results of this amazing study, I wanted to, again, start more at to deep dive more in the interpretation aspect of your results. For example, these findings, will you say, extend on what we know about maternal versus paternal neuroadaptations that we touched on a little bit with the example you gave, but it's directly is more, again, coming back to your study, you as authors discuss at all whether these findings, will you say, extend what we know about maternal versus paternal, or mother versus father neural adaptations or not, or TBD? And also, what might this study in general tell us about perhaps empathy that we've mentioned, or stress regulation, or even co-parenting dynamics?
Oh, yeah, all really, really good questions. So to answer the first question, the simple answer is this study is not poised to make that comparison between mothers and fathers' brain responses. We just didn't have mothers. However, what we're starting to see are, just if we kind of like took a step back and looked at the broad literature, we could start to make some guesses about kind of where those differences lie. It looks like overall, if you look at the longitudinal, admittedly small longitudinal literature, it looks like that mothers the changes that happen neuroanatomically for mothers is a little more pronounced than what happens in fathers. It looks like fathers, at least to our eye currently, that change is very much cortical, whereas like mothers, we see some changes both cortically and subcortically. Again, this is very preliminary. There's not been many studies, but just kind of taking a step back and looking, that's what we're saying about that adaptation. But in terms of like specifically what we did in this study, I think compared to mothers, since we didn't have mothers, I'll leave it at that. But in terms of the second question, so how do we think about empathy and co-parenting and all that good stuff? So
regulation, co-parenting dynamics.
Right. Well, I'll add one more thing about the findings, and I'll answer your question more directly. We did a follow-up analysis using multivariate pattern analysis, which really is just a fancy word for saying we wanted to classify certain categories of information based on brain activation using fancy machine learning, And basically what we found is that we found relative contributions for people that can classify a familiar stimulus and a baby stimulus, which is really important to kind of like validate our initial thoughts is that people have these unique responses to their own baby. It's not just about it being a baby, and it's not just about being familiar. We realize that both of those dimensions actually make significant above chance contributions. However, some of that chance or some of that contribution is unaccounted for, which we can imagine that there is something specific or something special about looking at your own baby. And what I think this means in terms of kind of all the other stuff you were referring to, empathy and all these things. I mean, the regions that we're talking about here, like I kind of mentioned this before, and this is an informal way to talk about this network of regions, this mentalizing network, but kind of like as a precursor to empathic concern or empathy, in the sense that these regions are supporting the ability to interpret others' feelings and thoughts. And these videos are videos of their infant who is nonverbal, which in real life, their infant will be nonverbal. They're not going to tell you they're hungry. They're not going to tell you they're thirsty. They're not going to tell you they're in danger. That's something that the parent has to infer or has to recognize based on other cues. And a lot of times it's going to be like a facial cues. The child looks really distressed, it's crying, whatever. So, I mean, the actual process, the actual mechanism we're talking about here is actually important for parents to develop, which is why, you know, we're so interested in these mentalizing structures. Simply because, yeah, they're supporting something that is incredibly adaptive, and without it, it would be tough to keep an infant alive.
Thank you for sharing that, because it makes me think on, and I don't know how more effectively communicated, but here we go. Listening to you, I'm also thinking, and maybe I missed it with your metrics, but how even all these findings you published may also change as a function of involvement, for example, parental involvement, which I'm not sure if something you guys, I know that you collected data on bonding, but if I'm not missing in my memory, all the metrics we've talked about thus far have been very related to assuming you are an involved father, but that may not be the case always. And
no,
therefore,
this is good.
Yeah, right? Because if we're talking ultimately how both behavior / experience and brain talk to each other, and both at the individual and then at the interactional level, if your experience in this case is not super engaged, how does that help or not in all these topics we've been talking that could be the regulation or the postpartum recovery, or how these longer term neuronal changes, so specifically neurofunctional changes that we see happen or not if you're an engaging, invested, caregiving father versus not, or when in the trajectory, because as we've talked, for sure is an interesting sample as compared to females. They don't go through the pregnancy phase. However, at the caregiving level, postpartum, the involvement that also happens there, you guys are demonstrating at the neurobiological level striking significant changes. But even within the fatherhood sample, how that may look like if your behaviors are differently. Have you guys at any moment considered that aspect, like fatherhood engagement versus not?
Yeah. Another disclosure moment for the paper. So actually in this paper, we did test that. I was going to say I put it in the small font. I did not. There's a sentence in there, but we did test the association.
The tiny one, the tiny.
Yeah.
But we are here expanding on that sentence.
But we did test that association between brain activation and the amount of time fathers spend as the primary caregiver, which is a bit more
minutes metric. Or hours? How did you
it's hours per week.
Hour per week.
It's an average hour per week metric, which you can, I'm sure people might think that's a little bit more of an objective, maybe measure of parenting experience. And this actually was not associated with brain activation to their own infant. Now, the caveat with all of this is we have an N of 32, for your audience, a sample size of 32. So it's totally possible that there are associations that we just missed simply because we didn't have the power to detect them. And, you know, that's no secret, and we're definitely not the final say on anything. However, it also is, it's an interesting point that we saw that these associations kind of arise in these self-reported metrics of bond and affective experience. We did not see this association arise when we looked at like the amount of actual time that the father was spending with the child. And just to give you something more to wrap your head around.
That's great. Well, I just, yeah, I missed that sentence. No, but that is good to know because, again, as I like to share with people, the science and the world, it is what it is. We cannot impose. We may hypothesize or we may expect, but at the end of the day, we get surprised by what the results are, and it comes back. Maybe it is not the case. It could be the sample size and therefore the power. And TBD, you guys will keep working. I hope that you guys keep measuring that because I think engagement, involvement, it's critical. And maybe you do find it, or maybe you do replicate that actually it's not an important metric. And I believe that in any case will speak stronger of how, regardless if you're so involved or not, like the experience in itself overall of being a father produces these changes and how it relates to the other metrics. I believe you want to expand.
I was just going to say quickly, just, I love everything you're saying. Just to say quickly, in our study, we didn't find that association. However, and if you go back to Darby and Magda's cerebral cortex paper, which was the first paper to track fatherhood changes across the transition to fatherhood from prenatal to postpartum, the neuroanatomical changes specifically, they found that brain structure, actually, the change in brain structure was associated with that time spent as primary caregiver. So whereas we didn't find this like cross-sectional association between brain function, or at least how they respond to their infant, and time as caregiver, we do see this like neuroanatomical kind of adaptation metric, which is structural gray matter volume decrease across the transition to fatherhood that's associated with that. So there's something there. And I think part of what we do as human neuroscientists, and I specify human because we're often not in the game of causal inference, or at least we're not that close to it. So as human neuroscientists, we get as close as we can, and we try to do as best we can, and we try to be as transparent as possible when we have findings. So there's something there, but I think that's where we're at now.
That's amazing. And good thing is that you're still very young in your journey, so you have many more questions that you'll be testing. And if I may, and I know that I've warned you about this, but I would love that we briefly touched, even if it's briefly, you as the second author, to also share with our audience a recent paper that also you published together with the first author, as well as all the other collaborators, in which, even though that we're not gonna deep dive, because it was led by Dr. Anthony Vaccaro, and hopefully in the near future we'll speak with him as well, it does totally relate, is very complementary to the study we've been talking, and I know that our audience will benefit a lot from learning at least the gist of T to get us going on how it relates. And for the audience, this study that was published pretty much around the same time that the study that Phil led was published is entitled Intrinsic Resting State Connectivity and Personal Meaning Across the Transition to Fatherhood. And it was published in the journal Emotion. And as I said, it really relates to what you were looking in the context of different populations of stimuli or stimulus, like baby, not your baby, and also a relational figure and the father himself. But if you can speak to us a little bit how that collaboration happened, because you're also an author, a very important one, like very higher up there, the second author. And how does it connect to your HBM work that you published as first author?
Yeah, great. So just briefly, so that Emotion paper, which was, as you mentioned, led by Anthony Vaccaro, who's now in North Carolina, essentially was the first paper in fathers that shows functional brain changes across the transition to parenthood in fathers. So that was something that was really, really important in itself. Now, one thing that me and Anthony share, and like our interest and our research interest, is our fascination with affective experiences in these major life transitions. And, we had this kind of measure of meaning making that we had been tracking, and we were able to kind of associate that with intrinsic functional connectivity, which for your audience is essentially a metric describing the functional organization of the brain at rest, essentially. And what we found is that these changes, that one, there was connectivity did change across the transition to fatherhood in interesting ways, but I guess perhaps more interestingly, it was related to change in meaning. And I should preface, actually, before we even get into that finding, that changes in meaning just in the sample overall was about split 50-50, which actually is really interesting, right? So some people change in the positive direction, some people change in the negative direction. So increased meaning and decreased meaning, which I think really highlights the individual experience that people go through in fatherhood. I mean, we try to make generalized assumptions, we try to run these group level analyses, but at the end of the day, like, there are these real differences, this is real heterogeneity across fathers. And I think it also just speaks to the fact that whereas meaning really, so when I think of meaning, it's like the thing that, so you can have positive feelings or negative feelings throughout fatherhood. You can be like, oh, I'm not sleeping enough, I don't feel good, I'm stressed. However, meaning is kind of that kind of central kind of construct that's like, well, I'm doing this in service of being like the best father I can be to this child, right? And for some people that really increases across this transition, and some people it didn't increase across this transition, which I think just really highlights the individual nature of how we make meaning across these major life transitions.
That's great. And in that said, what you already skipped one of my questions that was like debunking like the whole personal meaning aspect in the context of transitioning to fatherhood.
Mm-hmm.
So then diving into the actual findings, and thank you for having explained what functional connectivity is in a simple manner to our audience. Yep. The study did show that an increased functional connectivity, specifically in regions like the insula and the temporal lobes, specifically predicting higher postnatal meaning. So if you could share with us, what do these areas tell us about, for example, how fathers integrate both emotion and purpose?
Yeah, great question. So the insula, let's start there, because I think that one may be more familiar than the temporal pole, which is the other finding. But the insula is this kind of really, it's an emotion region. It supports kind of like the way we process emotions. And I think that when we think about this transition to fatherhood as this affective experience, I think we really made sense to us, actually, when we saw this finding, that this kind of core cortical region, the insula, that's really dealing or subserving kind of emotional processing, or how we're dealing with emotions, was predicting how basically fathers were able to make meaning out of kind of the task of fatherhood, or make meaning out of them being a father, because that in itself, baked into it, has so much effective information about what are the rewards or what are the positive things that I'm getting from my child. So I thought that was really cool to see that the insula popped up in that one. So the temporal pole may be a little bit less known to most of the audience, but really it's another social cognition region that's been implicated in mentalizing. Also one of the nodes of the default mode network. And I think this kind of echoes from what we saw in some of the other work that we've done, where there's this kind of deeply rooted social cognition adaptation that's happening in order to make parents good parents, in order to make parents the best caregivers, the best child rearers that they can be. Its association with meaning is also really interesting, because we think that this area of the temporal lobe is somewhat associated with like social realization. So I think figuring out kind of where you fit socially, kind of like in the scheme of things, I think that really changes across the transition to parenthood. Like, you go from like kind of being not necessarily on your own, but maybe like more like your focuses are more independent on kind of what you're doing, to this really sharp change of like, oh, I'm no longer like my motivations are no longer isolated to just me, myself, and I now have like this other thing I have to really worry about. And I think that's a huge identity shift. I think you have to really like kind of like look in the mirror and like, whoa, so I
I'm
think the
glad
findings were
you
cool.
I'm glad you spoke about identity, because actually I skipped that because we've been talking about different things. But one of the areas that I wanted to touch on, even from where we were talking about your lead study, but it integrates to this one. It's, I guess, with all of them together and all that you've learned thus far from this research you've been doing, it's, what will you say specifically what these neural responses from either study or both reveal about how fatherhood reshapes identity, if at all? Is that
Yeah.
something you guys have discussed? Is that off chart?
I mean, no, it's a good question. I think clearly, and I think Anthony's paper really is starting to touch that topic a little bit of like, how do we make meaning of being a parent? Or what is the protective role of like developing this sense of like fatherhood as an identity in order to kind of get through all the things that all the challenges that happen during the transition to parenthood. I mean, I already mentioned that. I mean, you sleep less, you're more stressed, you have higher rates of psychopathology. Like, why would anybody become a parent, right? Well, it's because I think one of the central or core constructs that drives it is it's meaningful. It's meaningful to be a parent. It's meaningful to take care of a child. And I think that also comes out kind of in that human brain mapping paper as well, is when we look at our own infant versus other stimuli that have similar characteristics. When we look at our own infant, there is something that's a bit different about that, and it's a bit different in these like really high order social cognition regions that are dealing with how we're able to mentalize, how we're able to understand or attend to someone else's thoughts and feelings. I mean, those are things that are so important, specifically to fathers, specifically to parents, excuse me, but specifically to being somebody who's taking care of a child. I mean, that's uniquely important.
Wonderful. And this relates to what I'm still curious, because you've brought up a couple of times your main interest, both now when you were, for example, highlighting what Anthony and you shared and leads to this collaboration, but even from your very beginnings when you were transitioning from undergrad to master's and figuring out your own pathway and including topic-wise, but you had this clarity that you were very interested in major life transitions. And in the context of what you've done up to now, what would you say these findings, again, from both studies or one of them, will you say help us, all of us, to understand adaptation? And also, kind of as we've been talking, not only in fathers, even though that has been the major sample in these two studies, but in anyone, in any human, going, as we've said, through major life transitions. How do you believe these findings help us understand adaptation in general across humans, if at all, or do you think it's very, very specific to fathers?
I mean, yeah, I think one thing that it highlights is, I think, especially in the developmental literature, you'll know this as well, is we are really focused on children. And that's not a bad thing. Children are very important. But the developmental literature has really kind of, that's where we've turned our heads. And I think studies like ours, or research programs like ours, really highlight the fact that the brain actually is pretty plastic throughout other like major transitions as well throughout the lifespan, not just in childhood, and also confer risks, like actual real like psychological risks as well. So I think kind of what I've learned as I've kind of gone through my research journey, and I think what people can kind of take away, is that we're always changing, and um, and I think studying the neuroscience, or studying the kind of the underlying biology of this change, is one, validates the experience on the one hand, but two, gives us a sense of where to point our attention when we think about policies and interventions and support systems during these major life transitions. So I think that's the biggest thing, is these major life transitions across the lifespan are accompanied by these neurobiological changes, it seems like. And I think to that point should be taken seriously and studied further for their actual kind of effects, whether it be mental health or otherwise.
That makes the perfect segue to the questions I have regarding more like your own reflections, your own learnings, and also future directions, both for you as a scientist and also the fields in general and in relation to policy. One of the many questions I've been pondering, even more getting more related to your work, is why, let's say from your standpoint at least, you believe is vital, is critical to include fathers in discussions about the neuroscience of caregiving and the neuroscience of family well-being in general. And also, if I may chime in, because for the audience, again, to learning is iterative, so I'll be repeating many things. In Phil's context, he's currently, and please correct me, but you're in a PhD of psychology, clinical psychology, I believe specifically.
Yep.
And for example, I did my PhD in neuroscience, and we all talk to each other and we work together to make things better. But for sure, each discipline also has its own uniqueness of contribution and methodologies, and brain health versus mental health gets typically confused. And in here, since you're in psychology, I want to also deep dive a bit more in mental health. So I guess we can start first with my question, but later we can deep dive in also what are, for example, misconceptions about fathers' emotion or mental health that you, as someone very involved in these topics, would like to see changes. But we can start first, again, with the neuroscience of caregiving and family well-being and why it's important to include fathers in that discussion.
Yeah, I mean, I think all that kind of, to me, got encoded as the same question, in the sense that, like, the things that we see in mothers, whether it be postpartum depression, lack of sleep, increased stress, we also see those things during the transition of fatherhood. They also have postpartum depression, like, for instance, and they also have increased risks of like high anxiety and high stress. So a lot of the psychological effects of parenthood, whereas I think they're very pronounced in mothers, they're also not exclusive to mothers. They also happen in fathers, and I think it's important to consider that in our scholarship. I think I was mentioning this a little bit earlier, but I think there's also just the simple fact that fathers are integral parts of the family system. We are unique in a sense as humans, to have this kind of allo parent fatherhood figure that is participating in caregiving. That's not, for your audience, that's not actually many species that are doing that. I think it's around like 5% of mammals have fathers that care for the child or for the offspring. So we're really unique in that sense, and I don't think we would have evolved in the way that we did with this fatherhood presence if it wasn't important for child development. So even if you're a developmental scientist that is really focused on children, it's hard to detach that away from father health or from parental health in general.
Thank you. And that's why I insist to the audiences, to the participants, to the community in general, that in the healthy bidirectional or in the positive bidirectional interactions that we find a lot of the health ultimately. In this context, more related to development, so typically mother, father, and kids, but I believe in general in the human experience, as you said, how much we need to always go back and look at our evolution, and we will find there the answers to our health, both in the context of already pathology, so how to improve it, how to treat it, and even more so in the context of health, how to optimize it. So thank you for these answers, and more related to you as a scientist that's still in this journey of learning, of constantly evolving and growing at the methodological level, for example, and also questions in general. I was wondering, you just published this, amazing. Also, as I tell people, when something gets published, believe me, I have 1,000 other projects going on, and I bet you do as well. So which kind of questions and or methods as of now, independently of these recent publications, are keeping you excited and you're looking forward to both achieve or try to complete in your doctoral work. But if it doesn't happen, as it typically happens, that we aim to cover all this in our doctoral work and half of it gets achieved, maybe in whatever comes after the doctoral work, what is keeping you excited in both right now and projections?
Yeah, we have just an interesting data set kind of in our lab that's hard not to be excited about. So much data that hasn't been published on. Like I said, we have this 7-year follow-up where like that same group of fathers that I published on in this study that we have prenatal data for as well. Those kids are now 7 years old, and we have fMRI data on those dads and their children. So, just kind of what things that I'm starting to think about is trajectory of changes across this kind of longer transition. So I know we were talking about before that really, really cool paper that came out. I think it was in Nature Neuroscience, where the group showed, I think it was Susanna Carmona's group or one of those groups, they showed this U-shaped trajectory of whereas like they saw neuroanatomical decrease kind of from prenatal to about birth, and then this recovery period after that. And that's something that I'm really interested in replicating in our fatherhood sample. I'm also really interested of also tracking changes in brain function across this kind of time scale as well, basically 7 years out. And then the final piece, full circle back to my post-bac with Danny Pine, is I want to see how the kids turn out. And I want to see like what are some of the things that were kind of some of the individual differences, things that were characteristics of the fathers and parents in general that are predicting child outcomes, whether that be in terms of their brain or in terms of their psychosocial kind of outcomes. So those are the things that are keeping me up at night, and essentially what my dissertations will be on.
That's very exciting, because that leads me to more like the nitty-gritty of methods. Again, what more broad are you as a scientist, that in the context of all those exciting questions, and also you already having learned and demonstrated a lot of very unique skills that we in neuroscience, and even more so if you are doing developmental cognitive neuroscience or computational cognitive neuroscience, you talked about MVPA, that not everybody is even familiarized. I appreciated you debunked it in a very brief way. I also did that when I was in my PhD, but it took some time to also understand what does that even tell you. In the context of your skills, how do you envision combining them? For example, your skills in multimodal imaging or longitudinal analysis, as you've been mentioning that now you're hoping to do this replication, but in fathers, or your computational methods that you also mentioned you've been playing with and publishing. How do you envision, if you've been envisioning this to combine them to keep expanding this field? And it doesn't need to be limited to your doctoral work.
Yeah, it's a great question, and something that I'm thinking about a lot, about how can I leverage kind of the most modern approaches to kind of squeeze the most juice out of the lemon being our data. So one thing that I think about a lot is multivariate analyses for neuroimaging data. I think that oftentimes we reduce or we limit our interpretation by taking a summary stat on a typical time point that kind of characterizes a group activation, and then seeing if that's different from the next time point to the next time point. And I think, well, that's great as a first step. I think a lot of times, especially if there's more subtle differences, which we might see in fathers, that these changes actually might happen across patterns of activation and throughout functionally distinct regions, not necessarily happening at this like kind of like group mean level. So that's something that I think about a lot in terms of how the brain is changing, and not just if the brain is changing. And I think also trying to, and this is a shout out to like kind of the department here. They've been really so helpful in kind of training me and continuing to train me on some of these methods, but like who can collaborate with kind of in other disciplines and other fields, and like how can we kind of bridge a bunch of different ideas, whether it be methodologically or otherwise, to make the most out of kind of what we have with our limited data.
I love it that you're already not only, of course, doing your doctoral work, but making the most of the resources, as you just mentioned, that you have at your local institution, making sure that your training is not only about this question, this paper, dissertation, and publish, but a lot of this PhD training process is also to see which other resources are out there so you capitalize on them to train, because even more so in the constantly changing world, us as scientists will end up in very different pathways that may not be the one that historically have been there for many decades, or even the ones that informed us when we were even younger and started this journey. So I love to hear that. And on a personal note, wrapping up kind of with both studies and all that we learned from it, I want to again go back to the human you and to learn if you've been reflecting on how this research has taught you anything about growth, for example, or purpose and connection, ultimately, again, not just scientifically, but humanly.
Yeah, it's funny you mentioned that. So I recently, not recently, last year, became a godfather. My best friend had his first kid, and it was around the time that I was actually working on some of these papers. And I think it's really nice to be able to kind of take a step back from like the nitty gritty details of your own work, and kind of seeing like it play out kind of like in your friends and in your family, and clearly with him. Like, I think there's so many like positive adaptations that I kind of see that he's went through simply just from the fact that now he's a father. So that was just, I mean, that's just like a very like salient, it was just a kind of a very salient experience for me, is having to kind of do this academic work, but then also at the same time kind of see like one of the people that's closest to me kind of go through it in real time. And I think those are the kind of experiences that, you know, you live for if you're doing human research, is seeing it play out in front of your eyes and not just in the data.
Yeah, I feel you. I tell people, like, we learn so much, of course, in the lab, and we're there for humanity to create knowledge, to put it in practice. But at the individual level, it's all the anecdotal, the experiential aspect. It's unique to complement all that we are as scientists constantly dealing with. But also when it happens to us in any of these different experiences, you also appreciate the what you tapped at some point, that the personalization, when you were talking about the half of the sample show this and half show that, like, also as much as we try to generalize, like there's a uniqueness in each experience. So
Absolutely.
that is wonderful. And to me, it's remarkable how your work really captures both the biological and the existential dimensions of fatherhood, as we've been speaking, showing that brain health is not only about neurons and networks, which are critical, but they also go in complementary or hand to hand with meaning and identity. So that's why I was also very excited to navigate these studies with you. So we see the synergies of our neurobiology together with our emotions, cognition, playing out all in this amazing human body, and in relation to the social interactions, which to me it really englobes the brain health um, experience, or umbrella, if you will say. So wrapping up, I would like to have one final questions for you, which will be if you could share with our audience one insight, mainly for future fathers, given that what you've been working on, and you guys have learned in your team, given what we know thus far, which is just scratching the surface. But if you're planning to become a father, what will you tell them is happening in their brains, and what would it be? Or even if you're just right now waiting, you're a couple expecting your baby, if
Yeah.
you can tell them in very simple ways what's happening in their brains, and what does it mean, or what would it be?
Yeah, it's a great last question. Um, I would say that your brain is changing to make you the best father that you can be. Um, and specifically, it's giving you, um, well, I won't say giving you, but it seems to be that it is supporting being more understanding, being more empathetic, being more ready to handle when something goes wrong, being more loving, and being able to withstand some of the real challenges that come with the transition to parenthood.
I love it. I'm very happy that you provided them with knowledge that you translated also into empowerment at the level of you can do this. And there's a lot of fear of both from motherhood and fatherhood that we sometimes just need to hear these words that from evidence and experience of many others, we can assure you that all is gonna be well and you're being modified, directly reshaped, but it's all for the good. So don't be scared. So thank you so much, Phil. I am so honored and grateful for your time because to me, this is a fascinating and generous conversation that our society needs. And I believe that your work together with your collaborators really deepens on what I believe is our understanding of how human connection directly reshapes the brain. So I tell people through behavior, in this case, human connections are is the direct pathway to reshaping both function and structure of your brain. And in the context of your studies, it also reminds us that fatherhood, equally or similarly to motherhood, is a profound neurobiological transformation. So thank you so much. And
Thank you for having me.
Of course, we look forward to many more future conversations with you and other scientists like you doing this amazing work. Thank you.
All right. Well, I appreciate it and take care.